Okay, so it's my great pleasure to introduce Dr. Victor Girza. He comes to us to neuroscience with a background in theoretical physics and philosophy. He did his PhD in theoretical physics at the University of Stuttgart. And then he started off his academic career at the Center for Complex Systems and Brain Sciences at Florida Atlantic University. So he spent several years there, and then he returned to Europe to accept a researcher position at the CRNS in Marseille. And he's still there today, and he currently serves as the director of l'Institut de Neuroscience des Systèmes. So he's a recipient of several awards, including the Erbsman Prize and the NASP-SPA Early Career Distinguished Scholar Award. He serves on many editorial boards and has published more than 190 scientific articles and book chapters, and co-edited several books, including one called The Handbook of Brain Connectivity, which is a really nice reference on sort of brain connectivity and how that is important in understanding the brain. He's one of the lead scientists on the Human Brain Project and the Virtual Brain. Dr. Jirsa has made important contributions to our understanding of epileptic seizure dynamics and also how network structure in the brain constrains the emergence of functional dynamics in the brain. So, without further ado, I will turn it over to him. We can get him up.
So, good morning, everyone. It's my pleasure to be here with you. It's a shame I cannot be with you physically. And I'm really sorry about that, and let's do the best. Let's make the best out of it. So I'm going into presentation mode, and
Yes, yeah. Please go ahead.
Yeah, okay. So Suen, by the way, Suen, hello, nice to see you. It was a long time that we've seen each other. And thank you for the very kind introduction. My talk is entitled When Causality Meets Inference, Digital Twins in Medicine. And what I wish to speak to you about is some of the work that has been done over the past, let's say, almost 10 years now. Much of it in the context of the Human Brain Project, which when I got back to Europe, I joined this effort, not right from the beginning, but in the second year. And what we have contributed to the Human Brain Project is actually an approach that is referred to as virtual brain modeling. In this context, we have developed a virtual brain neuroinformatics platform, in particular work that has been done together with Randy McIntosh and Petra Ritter. And I will give you an insight to what the essence of this approach is and how it integrates into the final deliverable of the Human Brain Project, which is a digital neuroscience infrastructure called eBrains, and to which I will give you an outlook at the end. Yeah, but with this digital neuroscience infrastructure, the endeavor is essentially to help neuroscientists, not just computational neuroscientists, but people wanting to use computational modeling to help to organize data, models, tools, and render them interoperable, and through this essentially increase the capacity of the community to move forward using this type of multi-scale neural model organization. And the challenges that we run into are actually that as a neuroscientist, your objective is typically to identify mechanisms that give rise to emergent brain activity that is hopefully linked to some type of brain function, or very often also to brain dysfunction. And in the brain, working with a multi-scale architecture, what we have is multi-scale always implicates actually the coexistence of multiple scales and certain scales feeding into others in order to compress some of the information that is in there. As a consequence, we are dealing with neurodegeneracy. Neurodegeneracy is essentially, in any multi-scale system, including the brain, it's the capacity of different mechanisms or different system configurations, different system arrangements or parameterizations giving rise to the same type of behavior. This is good because this provides the brain or any multi-scale system actually with the capacity of robustness against perturbations from the outside, because through plasticity then we can rearrange our internal organization in order to give rise to the same function. But it has, as a neuroscientist, when you're after the identification of a mechanism, you also have as a consequence the non-identifiability of these mechanisms, because multiple mechanisms can actually give rise to the same type of behavior. In machine learning, this is what we refer to as non-identifiability, and this is something we need to deal with, simply because it's at the essence of the nature of a multi-scale system, here in this case the brain. An approach to that is, if you want to disentangle mechanisms, you have to formulate a causal hypothesis. Yeah? So with pure machine learning or artificial intelligence, where you perform statistical inference that is extracting the structure that is in the data, you will not be able to do this. You must be capable, and this is neuroscience, of formulating a causal hypothesis that in the hands of most people is actually represented by a mathematical model. Yeah, and in this case, we're not talking about statistical inference anymore. We're talking about causal inference. That is the inference of the causal mechanisms in the evidence of the hypothesis that you put inside of your causal formulation and the data with which you confront the system. Yeah? And so we need a causal inference framework where we need to be capable of paraphrasing or formulating and operationalizing the multimodal framework, and then being able to use methods from machine learning in order to confront it with the data. There's one additional aspect that plays actually a very important role that I want to highlight here at this point already. When we talk about multi-scale, many of this type of information, when you want to go all the way to the patient, yeah, much of the information is actually available to us only ex vivo. In particular, high resolution information. As a consequence, when we want to apply this type of approach to patient data, we are losing aspect of individuality, because ex vivo data are typically coming from other sources. So we have to find a framework that allows us to take advantage on the one hand side from in vivo data in order to be able to perform individualized modeling in order to be able to go all the way to the patient. But then parallel, we need to, and that's orthogonal, essentially, we need to be able to find ways of integrating ex vivo data. And that leads us very naturally to hierarchical inference frameworks in which we can actually combine this type of data. I'll come to this later. What I want to do now, from having set your mind into the perspective of multi-scale modeling approaches being enriched by data and trying to put data, models, and methods, trying to render them interoperable, how can we do that? And for this reason, I would like to present to you the workflows that we have developed over the years on the macroscopic level, and I will connect it to the multi-scale level a little later on. Yeah. So what is referred to as virtual brain modeling is the following. You start with non-invasive brain imaging data, in particular structural data, for an individual subject or an individual patient that allows you actually to, on the one hand side, to extract geometric information. Very often, or generally, this is actually topographical information, individual reconstruction of the cortical surfaces and the subcortical areas. And with a diffusion tensor weighted imaging, we are capable of extracting what is today referred to as a connectome, that is the complete set of structural connections between individual brain areas. When we co-register this together in the same reference frame, then we have a skeleton, a framework that allows us to embed mathematical models. This is only in a form of an avatar. Yeah, it is a spatial representation thereof. So what we need to do in order to have these models for the individual brain regions, we need in order to render them active. Yeah, so they can actually communicate information between the individual brain regions. We need to be able to have a mathematics that allows us to capture the temporal responses to that. Yeah? And this is often being done on the microscopic level, Hodgkin-Huxley equations, for instance, for individual neurons. But this is not sufficient if you want to do this on a macroscopic level of a system networks level. And for this, what we are referring to is typically mean field models, where we are actually using individual information that is accessible to us about individual neuron functioning, connect these neurons, build individual populations, and then apply statistical techniques, so-called mean field techniques, where we extract the individual first, second, and up to fourth order momenta, yeah, mathematically, that allows us to reduce some of the dynamics into very few degrees of freedom, and also formulate the corresponding differential equations that prescribe the type of dynamics. Yeah, this is referred to as mean fields, or when you collapse it, then representing an individual brain region, it's referred to as a neural mass. Yeah, these are our individual network nodes. Yeah, and what you see here, and if not now, hopefully in 1 or 2, 3 seconds, you see a slide 7. I'm on slide 7, linking scales, some examples of how this is being done. Here, what I'm displaying is an example from Alain Destex's lab, a collaborator of mine in the Human Brain Project, with using so-called ADX individual neuron models that have been coupled with each other synaptically, where on the left you see raster plots for excitatory and inhibitory neurons. These are the differences in terms of the color code. Color in red is inhibitory neurons, that you can actually collapse mathematically using the statistical approach and build what is called an ADX network, an ADX population, and then the information that you can actually capture the same type of dynamics within the mean field equations. In the mean field equations, you have individual parameters that are actually collapsing and absorbing all the individual neuron characteristics that is coming. I'm hearing myself in echo.
Victor, it's Derek. My apologies. We are not seeing any slide changes here.
On which slide are you?
The first one.
The first one.
Correct.
I'm on slide 7.
There we go.
Are you on? Okay. I'm stepping out of presentation mode. Yeah. Can you hear me? Okay. Yeah.
I can. Yes. Thank you.
I'm pressed. I stay in this mode. Now you see slide 7.
Linking scales.
Yeah, okay, that's the slide. So I'm staying here. Sorry about that.
Okay,
the slides I just showed you, let's ignore them. Yeah, what I showed you was, do you see slide 5 now?
Workflows.
Okay, excellent, thank you. This is what I was talking about. co-registering information, topographic, geometric, and the topology leading to one reference framework, where you have then you build a network in which a node represents an individual brain region, which you then have to equip with the type of mean field dynamics that I just described on this particular slide here. To the left, you see the raster plots for different brain states, asynchronous state, slow oscillations as we see it in sleep states, and where the microscopic properties of the neuronal distributions, including the heterogeneity, are essentially collapsed into mean field parameters that allow you to have a low dimensional representation thereof. Yeah. So when we do this, then I'm on slide 8, the virtual brain. You can actually write down mathematically equations for coupled differential equations that on the one hand side have a nonlinear local dynamics that is representative for the isolated mean field dynamics. Then nearest neighbor interactions, which is typically local dynamics, intracortical, and the global dynamics, where you have communication via signal propagation between brain areas, in which in particular time delay plays a very important role via signal propagation along the fibers. Yeah, there I will not speak much about time delays today. There we are typically on the ballpark 100 milliseconds, and it's actually essential for the type of dynamics. Yeah. One aspect that is important is actually here we are on the source level. Yeah, and the source activity can then be mapped upon electrode activity or intracortical electrodes, EEG, MEG, or fMRI data using the corresponding biophysical couplings, like in fMRI, that's a balloon Windkessel model. So we are actually linking the source to the sensor space. Yeah. And what I have just described is available in here in bottom right, you see the virtualbrain.org. This was a platform in which we put all these elements together, allowing, and that was released in 2012. Yeah, it's an open source platform organized in or written in Python, in which you have all the pre-processing steps that are necessary in order to handle the type of connectivity, define a brain network model on this particular level, and then actually run the simulations, as you hopefully see at some point on slide 11, in which the activity evolves on the source level. Yeah. So you have dedicated stochastic and deterministic simulators to do this, or can and then from the source level you can map the activity also on the EEG, MEG, fMRI. So essentially on the sensor level, whatever your preferred sensor is, or sensor domain is, and then you having the type of activity as you observe it in the individual patient or subjects that you're dealing with. Examples that you see here is EEG data, where you can load in your EEG combination that you implementation that you have, or the setup that you use also in the lab. You can do this type of work also in other species. At the moment, as it exists in the virtual brain, you have the macaque in there, you have the mouse in there, and the mathematics that is behind there are 100% identical. The parameters are different, but the mathematics is the same. The good thing about that is it allows you actually to start testing hypotheses about what actually matters in this type of description in terms of network. On slide 13, you see a representation of probabilistic tractography of the mouse. On slide 14, you see the one deterministic tractography there. So you see you have much fewer false positive in this particular representation, but the fibers are significantly more sparse. On slide 15, what you see is representation of the Allen mouse connectome. This has been done in collaboration with the Allen Institute. There you lose completely individuality, obviously, because this type of atlas is being generated by the use of several hundreds of mice in which retrograde injections have been made, and you can retrace the individual fiber tracks. That is different for the DTI. Yeah, there you have the individuality. The question is, what is it actually that you're plotting? To what degree do you have false representations thereof? But within the virtual brain platform, you can actually now start quantifying this type of question and ask what does actually matter. And on slide 16, you see a representation thereof that has been some work that has been done by Melozzi et al. That was in collaboration with the Allen Institute, where you can actually test this type of workflows represented on the right hand side. So for instance, you can use the tracer data to build the virtual mouse brain and then equip it with the mathematics in order to generate resting state activity data in this particular case, and then map it upon the bolt signal to extract, then finally analyze it using the type of data features that you like them best. Here in this example, it's just FC, functional connectivity, which is a Pearson correlation of activity across individual areas. Yeah, you can do this also with the individual mouse data. You can use structural data in order to build the virtual mouse brain based on the individual structural data. Then you have an individual mouse brain. Yeah, and you can and then generate the functional data, or you can also measure the functional data and put the mice directly into the scanner as we do it with humans and actually measure the corresponding activity. This has been done systematically, and then you can actually, using various data features, compare. Yeah, and you can, and that's what you see on the left-hand side. You can also build different surrogates. You can, for instance, the Allen Atlas connectome data that are coming out of this, they are non-symmetric. What we get out of diffusion tensor weighted imaging is symmetric. So to what degree does actually symmetry play a role? Or there are many fibers missing in the DTI-based connectome. Yeah, what if we add them? Or what if we go into the Allen connectome and start removing the fibers that are actually missing in the DTI? This has been done systematically, and then you can actually perform the simulations and compare it against scan, rescan variability of the individual mouse itself. So you have it in the scanner for 20 minutes resting state activity, and then you scan it again. Yeah, and you can compare actually the predictive power that comes out of that. Yeah? If this is your point of comparison, the key message on this is essentially that you see the green tracer-based representation has the best and the biggest predictive power, and as you get closer to deterministic and probabilistic diffusion MRI, you see that the predictive power reduces. And when I go actually to the next slide, so systematically as you go through the individual steps, and when you quantify this on an individual mouse basis, yeah, you find actually that the high resolution data coming from the tracer studies have the best predictive power on the one hand side. Yeah, so this is ex vivo. In vivo, individual connectome still has a better predictive power than a generic connectome or taking a connectome from another individual. Yeah, so, and this gets us actually into the situation where we have ex vivo and in vivo data, both. One is high resolution, the other one is a low resolution but individual, that need to be put together into the same reference framework, leading to an increase of predictive power, and that essentially asks for a Bayesian hierarchical inference framework, which is the one that we're actually using here. I will get back to this a little later, but where we use essentially the individual connectome as a prior in the Bayesian inference, where we use actually a high resolution ex vivo connectivity coming from post-mortem data. And to give you a feeling for what the workflow looks like in applications, I would like to do this with you step by step in the example of epilepsy, and show you how far we can get with a regular framework before we go to the high resolution, and then show you the examples of what can actually be gained when we go to a high resolution brain reference framework, for which you actually need to have this merger of in vivo and ex vivo data. So a few words on epilepsy in terms of the workflows that we're using. One of the key questions in drug-resistant epilepsy is actually the organization of the epileptogenic zone. 2/3 of all epilepsy can be handled typically through pharmaceutical interventions, which means 1/3 are actually drug resistant. And for those patients, the only hope typically is surgery, epileptic surgery, yeah? In which you actually resect the epileptogenic zone, which is considered to be the primary zone of organization, very often linked to the onset zone of the epileptic seizure. In order to identify this, you typically take a combined approach of non-invasive imaging as a first step, and then invasive imaging as a second step, in which you implant electrodes. In France, something that is being done regularly is so-called SEG, stereotactic EEG, in which you insert electrode needles that have 10, 15, or 20 contacts that have a spacing of a few millimeters, but go into the volume, as opposed to electrocorticograms, where you actually have a grid on the outer surface. Yeah? With the SEG, it gives you the possibility to go in deeper structures and measure the activity that is actually occurring there. Currently, the success rate of surgery, well, after an application or hypothesized identification of the epileptogenic zone, is around 60 to 70%, typically in temporal lobe epilepsies. If you go outside of temporal lobe epilepsies, it can drop all the way down to 25% success rate. And it has not changed over the past years, over the past 30 to 40 years. It has improved, yeah. In particular, the technique has become under better control, but the success rate has only seen marginal improvement. Here, in terms of taking a causal modeling approach, we have the following workflow. Yeah, I'm on slide 20, by the way. Yeah, so we construct the virtual brain of an individual patient using non-invasive data and only structural data, yeah, in which we then span in three-dimensional physical space, so inside the head of the patient, virtually inside the head of the patient, we span a network after parcellation of the topography into individual brain regions and co-registering this with the individual connectomes. So here we have a low resolution representation, but take the individual connectome into account. Yeah, as a reminder, you saw that the beta predictor was high resolution, but this is what we do at the moment in routine. Yeah, in our research routine. As the next step, we equip it with mathematical models, the mean field representations. We bias the individual parameters if there is additional information, such as a lesion visible in the MRI. We bias the parameters in this direction, and then we are performing inference, yeah? Data fitting and model inversion by using a causal inference framework. I will go in more detail later on. Yeah. As a consequence, what comes out of this? So in this case, we are using the virtual brain as a target, as a formulation of a causal hypothesis in which we are balancing the parameters of the individual mean field model. One of them will be related to epileptogenicity, and the contributions of the structural connectivity. So in this balancing, there is an outcome which is linked to an estimation of the epileptogenicity in individual brain regions. And this is information that we are providing to the clinician. Yeah. And some of the mean field work, there has been loads of activity actually in order to build mean field models capable of generating epileptoform activity. And some of these models are mechanistic. Yeah? Many of these models have been actually formulated in a phenomenological form, matching the type of flows, the type of dynamic features as observed in the mechanistic model, but being represented here in a reduced mathematical formulation in which actually the parameters are more of generic nature, such as canonical models in terms of bifurcations. But that is nevertheless useful, because these models, we do not want to, the parameters, we do not want to interpret them mechanistically at this point, but we actually want to map out degrees of epileptogenicity. And what I'm displaying here are some of the mathematical papers in which this has been actually performed in much detail. Yeah? But what I want to emphasize here is the following. We have a non-stationary evolution when talking about the time course of an epileptic seizure. And what you see here is actually in tutor data of an extracted rat hippocampus, in which the activity was directly measured. On the left-hand side, you see a time course in blue. You see actually the neuroelectric activity as measured by the local field potential on a very long time scale. So these are multiple minutes of activity that are being plotted there. And in green, you see actually representative extracellular activity. It could be oxygen and ATP, but what I'm plotting here is extracellular potassium. This type of what you see here is actually that the extracellular potassium is actually tracing out some of the time course of the ictal activity. When you look on the right-hand side in picture B, you see a zoom in there. On the bottom right, you see on the time course of half a minute, this is a scale that is indicated on the bottom right, you see the evolution of the seizure, and as the slow variable actually traces it out, leading towards an onset and then going towards an offset. In this case, actually, the seizure was induced by putting the preparation into low magnesium conditions in order to render it epileptoform. Mathematically, you have this is a beautiful example of what we refer to as a fast-slow system, in which you can actually separate the fast subsystem, the fast variables, from the slow variables, and this was just one representative mechanistic representation. But in nonlinear dynamic system theory, you can actually formulate the existence of such fast and slow subsystems and formulate them actually in a timescale separation of coupled ordinary differential equations. Yeah? On the top right, what you see is actually a simulation of such a fast-slow system, in which you have these two timescales widely separated, but the fast time course, the fast activity actually determines the time course of the slow activity, which then itself again determines the fast activity. So you have a mutually coupled system, and you see the sequence as you saw it in the red data. you see actually the fast discharges representing the epileptic seizure, then the offset bifurcation that gets you back into the resting state dynamics, and then going back through an onset bifurcation into the fast discharge dynamic. When you actually take this representation and span it in what we call the state space, three-dimensional state space, you find actually this this spiral structure here on the left-hand side, where you have the resting state manifold. It's a slow manifold that is attractive, that has, as long as you stay on this manifold, this is the resting state activity, but you leave it at one point, which is where the part becomes unstable. You know? Think of the extracellular potassium as I showed you earlier, and then you go somewhere else in the state space, enter the spiral, and you get the fast discharges. This is a very generic, mathematically generic structure in which is characterized by an onset bifurcation and an offset bifurcation. This is where you actually leave the resting state manifold to go into the spiral, and how do you leave the spiral to go back into the resting state manifold. Yeah, so we can actually mathematically formulate this, and actually we can formulate this rigorously, and we can actually characterize it by a set of onset and offset bifurcations. If we are content with concentrating ourselves on so-called planar bifurcations, then we can actually formulate this in a finite manner, because there is only a finite number of ways of how you can enter from resting state into an oscillatory activity, and vice versa, how you can leave it. And that allows us to build actually a taxonomy. As Izhikevich has done that previously for bursting neurons, we can actually do the same thing for mathematically the same for epileptic seizures, characterized by an onset bifurcations versus offset direct bifurcation, and build individual pairs that allow us to systematically classify that and build this taxonomy. And this has been done systematically, and actually now I'm not in presentation mode. I need to move this here. Tested in seizures in a collaboration with Bill Stacey and Christophe Bernard, with 12, 13 other laboratories in Australia, Japan, in the US, multiple laboratories in various European countries. More than 2,000 seizures were looked at and actually were systematically classified in terms of their bifurcation features, bifurcation characteristics for onset and offset. Here you see just two examples coming from data. The characteristic of this data was that they were actually performed with DC recordings, and clinical seizures are typically performed with AC recordings, but there you're losing actually some of the features that are linked to what we refer to as a baseline jump, when you jump from the resting manifold to the oscillatory manifold, and we wanted to extract this. When we do this systematically, what happens is actually we can get statistics that allows us to identify the different types and classes that you find. And it turns out that there are two dominant classes. Yeah, for the onset bifurcations, it's either the saddle node bifurcation or a hop bifurcation, and for the offset bifurcation, it's a homoclinic bifurcation or another hop bifurcation. Yeah, so these combinations you can build, and that allows us actually to build a classification scheme in which we are able to systematically parameterize the system and actually make predictions how the system varies in parameter space as the slow variable guides us through the parameters. And that is actually very important, because it provides us, when we build an individual's virtual brain network, we have priors of the individual's parameters as a function of the seizure type. So that provides us a first primary focus. Once we have done this, then we take the individual's virtual brain, and actually we need to pass it through the inference framework. And for this, we need to actually identify a data feature or a set of data features. And there are different ways of doing this. And the way we are doing this is we are using actually the envelope functions. Here on slide 26, you see an example of multiple seizures coming from multiple patients. On the right-hand side, the red curves are actually representing the envelope functions that we are actually using to fit for an individual's seizure envelope, yeah, which is then characteristic for the individual parameters. We pass it through our inference framework, where we are actually using a Markov chain Monte Carlo framework. We are sampling the parameters that give rise to exactly this type of envelope function. And as a consequence, what we get out of this is actually parameter distributions, so-called posterior parameter distributions. And because we are sampling them, we're not just fitting them, but we are sampling all possible different realizations, leading to the same type of propagation pattern as captured by the envelope functions, as you have seen here. Yeah, we are getting actually much more information. Remember, I told you about non-identifiability. This happens when you have multimodal distributions. And hopefully we are on slide 28, in which I'm showing you some of the examples that are coming from experimental data leading to this type of estimation of posterior distributions. On the top right, you should see a representation of a multimodal distribution. One of the parameters that we are fitting there is the epileptogenicity that actually gets the system away from the resting state manifold towards the spiral attractor. So this is a degree of stability that is being interpreted as epileptogenicity. And this, for one brain area, you see represented on the top right on slide 28. And with the Monte Carlo approach, we are actually sampling the multimodal distribution. Yeah, if we perform a different type of approach in terms of optimization, or if we so for maximum a posteriori approaches, so there's another type of approach that provides you with a posterior distribution, but actually estimates the maximum of a multimodal distribution. This is where the green cross is oriented. This is something you can do, which actually reduces the computational needs in order to perform this. The Monte Carlo approach, the MCMC approach, is the most demanding of this, but it's the most precise also. So if you have, there are other approaches such as variational inference, yeah, in which you make assumptions about the distribution of the posterior probability. Like here in this red case, this would be variational inference. This is what we do in dynamic causal modeling, for instance, there in DCM, and there you can actually invert it very fast, but sometimes it works, sometimes it doesn't work. And here I chose an example just as a red flag to point out, look, this is actually experimental data. Multimodality is a reality. Yeah, and multimodality in this particular means that the explanatory value, the predictive power of a low degree of excitability or a high degree of excitability has actually the same degree of predictive power. We cannot be certain. We have to take both combinations into account. Yeah, this is what you see on the top right, and the way that translates then is because what we pass on to the clinician is distributions, yeah, of individual parameters, which you see represented on the left without structural connectivity and with the structural connectivity. And what you see there are for different brain areas. You see different violin plots, which are the posterior distributions. And if we do not take the structural connectivity, the connectome I was talking about earlier, into account, we actually get different results. And this here is actually simulated data. In this case, we know the ground truth. we get the wrong results. Yeah. So the structural connectivity plays a very important role. Why is that important? Because what is entitled here in terms of real world scenarios, what we are passing on to the clinician is actually the target for the surgery. Yeah. So which is being taken into account in the decision making. Yeah. So here 2 examples for actually on slide 30, 2 examples where you see what we refer to sometimes as a heat map. The heat map, so in red represented what we have deemed to be high epileptogenicity values, and in the upper row you see an example with a good overlap with the resection that has actually been performed with a seizure-free patient. What you see there is a post-surgical MRI. Yeah, so the black areas, this is actually where the resection has been performed. Underneath, you see an area, you see an example from a resection in a non-seizure-free patient, where actually the heat map does not correspond to the surgery that has actually finally been performed, and this patient actually turns out to be non-seizure free. And this is then actually the type of outcome that we used for evaluation of actually the success of the prediction. This is being tested in a clinical trial that is ongoing now since 2019. There are 13 clinical reference centers that are actually contributing to this trial in France, where the patients, about 400 patients, are being included in the trial since June 2019. The patients, the data, all the data that we need for the virtualization are being collected. They are being anonymized in a central database in Lyon, then being sent to us in Marseille. We perform the process of virtualization and go through all the steps that I have shown you, identification of the dynamo type, reconstruction of the structural connectome on the structural avatar for the patient, rendering it active, yeah, through equipping with the mathematics of the individual brain regions, introducing all the biases for the priors, and then going through the process of estimating the heat map, which is then being sent back to the individual decision-making staff that for half of the patients that we are receiving is taking it actually into consideration. Yeah, the trial, of course, like everyone else, got slowed down through COVID. Nevertheless, at the moment, on slide 32, December, we had 210 patients included. Today, we have approximately 250 patients. So there will be a delay for the outcome of the trial by approximately a year. In 2023, we should have statistical numbers that we need. Some primary results for retrospective data when compared to retrospectively. So here it was not prospectively taken into account, but retrospectively we can take some patients and evaluate how they have been operated and then compare it against the virtual brain prediction. And you see in this example where precision, statistical precision, is being mapped over CH stands for clinical hypothesis and VEP stands for virtual epileptic patient. So for the seizure-free patients, we are definitely matching the quality of this current state of the art. And for the non-seizure-free patient, you see actually that it drops down significantly, statistically significantly, indicating that there is a conflict with what has actually been performed. Yeah. And on slide 33, you see essentially performance for about 53 patients using a large range of different performance measures that are being used for the evaluation when you compare essentially the heat map against the type of surgery that has been performed. So in this case, only the virtual brain, the actual capacity of generating seizures has been used only as a target for model inversion and for the inference of the epileptogenic zone. We can do more though, yeah? We can actually, for every single patient, we can go inside and actually start asking questions about what would have happened if we had resected a particular area, or if we had disconnected the brain. So we can, the brain regions that are part of the epileptogenic zone. And this is much of the activity that is ongoing right now in terms of taking full advantage of the generative power of virtual brain modeling, because you can use it actually as an in silico platform to ask this type of questions, what if? Yeah. So not just using it as a target to estimate parameter distributions, but actually take advantage of its capacity of generate individual brain signals. And evaluating, for instance, different surgery strategies, yeah, because there are also other strategies than resecting an individual brain region. What you can do is you can, for instance, disconnect individual brain regions and still keep the tissue in the brain, yeah, which has many advantages, in particular also simple mechanic advantages, because you don't have a hole in there in which the tissue is then missing. Yeah. So I do not have the time to go into this, but I wanted you to be aware of this aspect of generative modeling. What I wanted to do in the remainder of the time is actually give you an idea of what can be done when we are actually going into the direction of not only using individual data, but now putting it into a framework of a high resolution framework where we use also ex vivo data. And this is something that is being done within the Human Brain Project and available in eBrains. Yeah, so this digital neuroscience environment, neuroscience, neuroinformatics platform that has been made available now, that has been launched 2 years ago, actually, and it is, if you want, you can say that this is the primary deliverable of the Human Brain Project. It is accessible for everyone. All you need to do is to request an account. It is accessible freely for the individual researchers with time allocations. And essentially everything that I described right now and much more, and also will describe in the remaining minutes, is essentially integrated as workflows available to the individual researcher or individual laboratories. And essentially, we should be on slide 43. Essentially, as a researcher, what you see displayed here is an abstraction of the inner functioning. On the top level, you see different operations that as a researcher you would like to perform. You would like to have data that you connect, put them in an inference framework, build a model for either an individual or very generic. So have actually data that are supporting structural data that allow you to build an individual microcircuit, yeah? And then you define your level of characterization. You want to define it on the individual region level or on the individual neuron level, or you want to have a mixed representation where you actually want to go for the hippocampus in much detail and all the other regions are satisfactorily represented using some mean field modeling. So essentially, as a scientist, you have very well articulated questions, and at the end of the day, you don't care how it's implemented in the hardware and in the software, as long as it's being done correctly. Yeah, so this is actually what is represented on the bottom level. You can ask what is under the hood. This is the idea of the eBrains, that you actually enable the scientist to formulate this type of workflows that you want to execute. Yeah, and what we are doing is we are offering to do this actually. My team is using mostly notebooks, yeah, Jupyter Python notebooks. There is much flexibility because you can actually access the individual modules that we are talking about. There are graphical user interfaces that are currently in production and that will be available soon. What is under the hood is essentially multiple supercomputers, in particular the so-called Phoenix research infrastructure that is available, on which actually the software runs. The software is composed of multiple simulators that have been rendered interoperable, data that are available, that are accessible through a knowledge graph that can be actually interrogated. This is what is called Zebra here. It's actually an API that allows you to navigate from the knowledge graph to enable the finding of data and then map it into a atlas reference space in which you can actually organize the data and map them directly on the individual model parameters on the scale of operation that you want to operate on. Yeah, and there are many protection issues, which is identified here as health data cloud space. If you want to put in human data or you want to access some of the human data in there, that is possible, and many data protection issues need to be addressed in order to make this possible, but this is work that is ongoing and is actually being addressed. In this framework, it becomes possible to ask actually the questions about the higher resolution. And one way, or the way we are doing is, for instance, is using the big brain histological space, in which actually histological slices with a resolution of 20 micrometers have been performed in Katrin Amund's lab together with Timo Dickscheid. This space has been defined as a reference space in eBrains. We are using it. With the work from Jean-François Mangin, it allows us to actually perform nonlinear spatial transformations to other spaces, such as the M&A space, with in this case 1 millimeter resolution that allows us actually to map data features that are coming from different sources and represent it all in the same brain reference space. Here in this example, you see actually Roxana Koymans working on a donor brain that has been received, that is actually being investigated in multiple laboratories, one in Amsterdam, one in Paris, the other one in Jülich, where actually histoimmunostaining is being performed, providing us with protein data that are being represented in the same reference space. Or on the right-hand side, you see an example of polarized light imaging, PLI data that allow actually a reconstruction of fibers of this particular brain with a micrometer resolution, providing us with a near to cellular resolution. Yeah? An example I would like to show you what we are using, what is already in use, is an example what is referred to as a Chenonceau brain, where we are having actually post-mortem high resolution imaging data on a resolution, this coming from Cyril Poupon in Paris, 200 micrometer scale. The connectivity on the right-hand side, we are on slide 47, yeah, is being represented in the same reference frame, in this case, mapping it on what is coming out of an individual's geometric representation. There we are on the 1 millimeter scale, and we represent it together in the same reference frame. This high resolution allows us actually to perform significantly better mapping from the source space to the individual sensor space. So going back to the example of epilepsy, here on the slide 49, you see a representation of one of the SEG electrodes, what I showed you for the patients earlier, that we are taking only the distance information into account because we do not have a correct definition of the orientation of the dipolar momenta across to the surface, because when we deal with a resolution of about 200 network nodes, the average brain region area is about 10 square centimeters. Yeah, this is simply not good enough. When we are going to the high resolution brain, here in this example of Chenonceau, there we are at a resolution of 200,000 vertices that provides us a spatial resolution of about a square millimeter, and these are the vertices, and that allows us actually, this you see in this example, the representation of very detailed neural fields and performing a mapping of the electromagnetic forward solution from the SEG contact towards the source distribution across the cortex. And this is subcortical areas. And this is actually very important because now we can actually describe phenomena as here in this example from Cathy Chavon's lab, actually, where you have real patient data measured on the one hand side with the ECOG, simultaneously with microelectrode recordings, yeah, and you have actually the multi-scale, beautiful multi-scale organization that is being described there and actually measured realistically, and you see that there is a rich dynamics on both scales of organization. Now with a multi-resolution, multi-scale representation here with the neural fields, this will become possible. Just as an example, on slide 51, there I'm showing you on the left-hand side essentially the fibers that come out of the rhinal cortex, yeah, stay intrahemispherically, but map also on the other hemisphere, yeah. And if we are taking in this example the epileptogenic zone just in the rhinal cortex and start increasing it once we equip it with the appropriate mathematics using the mean field equations we spoke about earlier, but now at the high resolution scale, what can be essentially done, I hope you can see this, a movie is running now where you have actually activity that is being generated in the rhinal cortex, and first it starts projecting onto some other further fibers, but then it starts propagating intracortically across the cortex, exactly as we have seen it in the example of the real patient data, in which actually both phenomena on the local, very high resolution level and projections into other brain areas, this is actually happening. So this is becoming possible. Another element that becomes possible now is this, what you see on slide 53, is actually we can now map the hippocampus as a continuation from the cortical sheet, because it maps, it's actually continuous along and starts developing in individual foldings, yeah, and into the CA1, CA2, 3, 4 regions, yeah, and we can have a continuous, spatially continuous continuation thereof, and activity that propagates through the cortex can actually go all the way into individual regions of the hippocampus, yeah. So this becomes possible at this level of a resolution. What can be done in parallel now is when we have this millimeter resolution, we can actually now connect the individual simulators. And on slide 56, what you see here is an image of the co-simulation technology. As I already implicated earlier, we can take a region of interest of, and in this case, it's a CA1 region of the hippocampus that we have folded into the cortical continuation of the cortical sheet, and we can actually extract data of individual neurons and actually perform a high resolution representation of the CA1 region and actually simulate the rest of the other regions using the mean field representations with the virtual brain. And this, just as a proof of concept, can be done. And in slide 57, you see a representation thereof where we use the data from Javier de Philippe in Madrid, who has performed some electron microscopy and imaged essentially the three-dimensional positions in the human CA1 hippocampal network, yeah, extract the positions of an approximate 14 million cells and represent them in three-dimensional physical space. The connectivity can be reconstructed using, for on individual cellular level, the probability clouds, computing the probabilities of synaptic connectivity, and using some electrophysiological recordings of human hippocampal slices. In this case, it's data provided by Hoyps Mansfelder. We can actually compute the individual electrophysiological response functions and allowing us to map the response characteristics on individual point neurons and actually collapse that. And then the type of simulations can be performed that you see represented here on slide 58. On the left-hand side, you see the CA1 in full isolation and using the 14 million cells just within a nest simulation, and on the right-hand side actually embedded into a larger scale context using the full brain network, which changes actually the dynamics completely within the high resolution framework in itself, also generating the type of propagating activity across the individual cells and organizing itself. So this type of work is becoming possible as of now, and that's the part that I wanted to share with you in order to give you an impression of what can be done in the eBrains framework as a proof of concept. And the idea will be essentially that this shall lead us to a capacity in terms of vision for the long term. We are still far away from engineering validity when talking about digital twins in engineering sciences and manufacture on the industrial side. The digital twin technology does exist, yeah, and there it represents essentially a virtual object that represents the physical object, but data being collected from the in the real world and that are being merged with a model. And on slide 60, you see this actually in a representation. This is coming from the NASA, this on the NASA roadmap, this image that I'm showing here, where they are actually using this type of digital twin technology in order to mimicking data coming from flows that have been measured experimentally. What we are aiming for in the eBrains framework is for the long run that we are essentially able to build under certain conditions on a chosen level of scale, because we have a multi-scale system here. Also virtual brains or digital twins of individual brains or generic brains, depending on what your question is, with a multi-scale representation, and then introducing data such as stimulation data or contextual data, visual, auditory data, and actually driving the model, and again linking empirical data with the model data, performing causal inference, perform estimations with the goal to actually to then narrow down questions, go across the multiple scales, with the regions of interest that require a more detailed microscopic representation, as I showed you in the last example, allowing to formulate this. Yeah, that would be the vision for the long term. We are still far away there. Yeah, however, eBrains offers a possible infrastructure to even at least pose this type of question and then see how far we can get. This as a vision was the last part that I wanted to share with you. And I'm essentially stopping here. I'm thanking my colleagues in France that are listed here that have contributed to much of the work. There are many others within the context of the Human Brain Project, and I wish to thank the funders there that have enabled this over the past, well, decade, essentially. Thank you very much for your attention.
So there's this question. You said that there are delays in the brain about 100 milliseconds. Do we know what the diameter of the brain's network is, the maximum number of synapses between two neurons?
Well, when you look at these fibers, yeah, they have, you can look at the fiber distributions and you can attribute to different regions and to different type of connections, different fiber lengths. We can reconstruct them. So between the fibers that are represented there, the longest ones are in particular the ones that are crossing the hemispheres, so the interhemispheric fibers, and they reach up to 20 centimeters. Yeah, so there we are talking about fibers connecting brain regions with one single synapse, but the fibers reaching about this type of distance. You find fibers, when you compare the fibers that are remaining intrahemispherically, they can also be very long, but it's the ones that are interhemispheric that are the longest. Yeah, so we are talking about one single synapse connecting individual areas. And actually, when you look at this, probably it's true that the biggest mass of white matter is actually in the callosum, but it is also those areas that are showing the largest degree of neurodegeneration during healthy aging. So I'm not talking about pathological aging, yeah, but when you look actually, and there is significant evidence in the literature on this, fibers that are crossing actually from one hemisphere to the other, that the largest degree of degeneration in terms of white matter you are finding actually there. Yeah, so fiber length matters. Fiber length depends on which areas are connected. And in terms of healthy aging, so no pathology involved whatsoever, it is one of the biggest contributors and determinants for the changes in the brain and associated not only brain function, but also associated cognitive capacity.
Thank you. We have Nicole asking, thank you for the talk. E-brains seem like a very powerful tool. Has this framework been used for studying Alzheimer's disease?
People are working on this. This is actually a great question. People are working on this. In my previous answer, I have already spoken about healthy aging, Alzheimer's disease. There are multiple labs working on this. There are still issues in terms of putting patient data onto e-brains and making them accessible, but I alluded to this earlier in terms of data protection, but it's being moved forward. So there are already data sets that are available and integrated, partially integrated, definitely in terms of structural data. And people are working on this, in particular, trying to map out individual changes and follow them longitudinally. Example is distributions of A, beta and tau deposits as visible in some brain imaging data, and then trying to link it to representations of using the type of technology I demonstrated today in terms of epilepsy, trying to adapt this in terms of neurodegenerative diseases. One of the big challenging aspects there is what are good metrics to characterize the data features in the brain imaging data. It's not by coincidence that I have spoken to you about epilepsy, because epilepsy as a dynamic disease, there are very visually speaking, or visually accessible data features. This is much more difficult in other diseases, in particular pathological aging, as in Alzheimer's disease. So there many questions arise already on an earlier level. However, questions can be asked causally. For instance, you can implement hypotheses about the various, it's called referred to as the Brack and Brack stages, the different stages during the development of Alzheimer's disease. And what some of the laboratories are doing are they are actually mimicking these different stages in order to generate then virtual brain imaging signals in order to guide questions about the data features. So it's early times, but people are using it for Alzheimer research.
Thank you. The last question we have here, you mentioned that probabilistic models have lower predictive power, but the mean field model includes a noise component, which is presumably stochastic. Do the deterministic models exclude the noise component mentioned in the mean field? Also, does the relatively lower predictive power of probabilistic models imply that causal interference models, Bayesian or otherwise, which are probabilistic, suffer from the same problem in comparison to deterministic and or tracer models?
Yeah. So we have to be careful there, or to properly distinguish the definitions. One aspect is I have referred to probabilistic tractography. Yeah, this is a reconstruction, a form of tractography to reconstruct the connectivity. When we so there, it turned out to have the lowest level of predictive capacity, probably to the large number of false positives that are being found in this reconstruction approach. Yeah. Deterministic tractography has demonstrated a better predictive power. Yeah. So this is on tractography. What comes out of tractography is a set of tracts that provides a connectivity matrix in the network models. Yeah. This has nothing to do with the type of mean field modeling. So out of the tractography, either one technique or another, we get a connectivity matrix, or we get it from ex vivo approaches as in the unconnectome. So this is a connectivity matrix. Yeah, these are, now we build a network. In the network is defined by a set of nodes plus a connectome, so a connectivity. Yeah. These two we put together, and we can span it in three-dimensional space inside the head. What is coming then next is the mean field models. Yeah, so that's now a completely different story. The mean field models render the tissue active in the sense that you do not have only passive tissue properties such as the distribution of matter that can be taken into account in the physics types of models that you have in there to link source to sensor signals. All these are passive properties. Yeah, we are rendering the tissue now active by introducing the mean field models. Those are also probabilistic models. Yeah, in the following sense, when we go from the single neuron model to the mean field, the mean field calculation actually performs some averaging over some of the probabilistic aspects, and then the mean field equations become fully deterministic. In this sense, we lose some of the probabilistic properties, and I assume this was behind this question that was asked. Yes, true. But then we are adding a form of phenomenological noise into the mean field equations. So we are not deriving the noise, but we are actually adding a form of phenomenological noise that actually renders the mean field equations now probabilistic themselves in order to essentially capture all the aspects that we did not capture correctly. And there is a certain, there is loads of variability and noise on this, yeah. But this we're not able to derive due to the mean field approach. This is being done phenomenologically. I suspect that this was behind this question in terms of mean fields. If not, please feel free to write to me if I misunderstood the question. I'll be happy to answer in writing.
All right, let's thank Victor Girza one more time. Thank you.
Thank you, everyone. Yeah. Bye-bye.